Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Ribosome. 91 languages. Large, red, and small, blue. Subunits of a ribosome. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. Ribosomes, /ribosome, so m / are macromolecular machines, found within all cells, that perform biological protein synthesis, messenger RNA translation. Ribosomes link amino acids together in the order specified by the codons of messenger RNA molecules to form polypeptide chains. Ribosomes consist of 2 major components, the small and large ribosomal subunits. Each subunit consists of 1 or more ribosomal RNA molecules and many ribosomal proteins, are proteins. 1, 2, 3, the ribosomes and associated molecules are also known as the translational apparatus. Overview, edit. The sequence of DNA that encodes the sequence of the amino acids in a protein is transcribed into a messenger RNA, mRNA, chain. Ribosomes bind to the messenger RNA molecules and use the RNA's sequence of nucleotides to determine the correct sequence of amino acids needed to generate a given protein. Amino acids are selected and carried to the ribosome by transfer RNA, tRNA, molecules, which enter the ribosome and bind to the messenger RNA chain via an anticodon stem loop. For each coding triplet, codon, in the messenger RNA, there is a unique transfer RNA that must have the exact anti-codon match, and carries the correct amino acid for incorporating into a growing polypeptide chain. Once the protein is produced, it can then fold to produce a functional 3-dimensional structure. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 13 centrosome. 14 cell membrane. A ribosome is made from complexes of RNAs and proteins and is therefore a ribonucleoprotein complex. In prokaryotes, each ribosome is composed of small 30s and large 50s components, called subunits, which are bound to each other. 1. 30s has mainly a decoding function and is also bound to the mRNA. 2. 50s has mainly a catalytic function and is also bound to the aminosylated tanners. The synthesis of proteins from their building blocks takes place in 4 phases: initiation, elongation, termination, and recycling. The start codon in all mRNA molecules has the sequence August. The stop codon is one of UAA, UAG, or UGA, since there are no tRNA molecules that recognize these codons. The ribosome recognizes that translation is complete. 4. When a ribosome finishes reading an mRNA molecule, the two subunits separate and are usually broken up but can be reused. Ribosomes are a kind of enzyme, called ribozymes because the catalytic peptidyl transferase activity that links amino acids together is performed by the ribosomal RNA. 5. In eukaryotic cells, ribosomes are often associated with the intracellular membranes that make up the rough endoplasmic reticulum. Ribosomes from bacteria, archaea, and eukaryotes, in the three-domain system, resemble each other to a remarkable degree, evidence of a common origin. They differ in their size, sequence, structure, and the ratio of protein to RNA. The differences in structure allow some antibiotics to kill bacteria by inhibiting their ribosomes while leaving human ribosomes unaffected. In all species, more than one ribosome may move along a single mRNA chain at one time, as a polysome, each reading a specific sequence and producing a corresponding protein molecule. The mitochondrial ribosomes of eukaryotic cells are distinct from their other ribosomes. They functionally resemble those in bacteria, reflecting the evolutionary origin of mitochondria as endosymbiotic bacteria. 6, 7. Discovery, edit. Ribosomes were first observed in the mid-1950s by Romanian-American cell biologist George Emil Pallad, using an electron microscope, as dense particles or granules. 8. They were initially called Pallad granules due to their granular structure. The term ribosome was proposed in 1958 by Howard M. Dintsis. 9. During the course of the symposium, a semantic difficulty became apparent to some of the participants. Microsomes mean the ribonucleoprotein particles of the microsome fraction contaminated by other protein and lipid material. To others, the microsomes consist of protein and lipid contaminated by particles. The phrase microsomal particles does not seem adequate, and ribonucleoprotein particles of the microsome fraction is much to explore. Our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Ribosomes assemble polymeric protein molecules, the order of which is controlled by the messenger RNA's molecule sequence. Ribosomal RNA composition for prokaryotes and eukaryotes. Awkward. During the meeting, the word ribosome was suggested, which has a very satisfactory name and a pleasant sound. The present confusion would be eliminated if ribosome were adopted to designate ribonucleoprotein particles in sizes ranging from 35 to 100 s. Albert Claude, Microsomal Particles and Protein Synthesis, 10. Albert Claude, Christian de Duve, and George Emil Pallad were jointly awarded the Nobel Prize in Physiology or Medicine, in 1974, for the discovery of the ribosome. 11. The Nobel Prize in Chemistry 2009 was awarded to Venkatraman Ramakrishnan, Thomas Stitz and Ada E. Yonath for determining the detailed structure and mechanism of the ribosome. 12. Structure, edit. The ribosome is a complex cellular machine. It is largely made up of specialized RNA known as ribosomal RNA, rRNA, as well as dozens of distinct proteins. The exact number varies slightly between species. The ribosomal proteins and RNAs are arranged into two distinct ribosomal pieces of different sizes, known generally as the large and small subunits of the ribosome. Ribosomes consist of two subunits that fit together and work as one to translate the mRNA into a polypeptide chain during protein synthesis. Because they are formed from two subunits of non-equal size, they are slightly longer on the axis than in diameter. Prokaryotic ribosomes, edit. Prokaryotic ribosomes are around 20 nm, 200 A, in diameter, and are composed of 65% RNA and 35% ribosomal proteins. 13. Eukaryotic ribosomes are between 25 and 30 nm, 250-300 A, in diameter with an RNA to protein ratio that is close to 1. 14. Crystallographic work, 15, has shown that there are no ribosomal proteins close to the reaction site for polypeptide synthesis. This suggests that the protein components of ribosomes do not directly participate in peptide bond formation catalysis, but rather that these proteins act as a scaffold that may enhance the ability of RNA to synthesize protein. See ribosome. The ribosomal subunits of prokaryotes and eukaryotes are quite similar. 17. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Molecular structure of the 30S subunit from thymus. Thermophilus. 16. Proteins are shown in blue and the single RNA chain in brown. The unit of measurement used to describe the ribosomal subunits and the RNA fragments is the Svedberg unit, a measure of the rate of sedimentation in centrifugation rather than size. This accounts for why fragment names do not add up. For example, bacterial 70S ribosomes are made of 50S and 30S subunits. Prokaryotes have 70S ribosomes, each consisting of a small 30S and a large 50S subunit. E. coli, for example, has a 16S RNA subunit, consisting of 1,540 nucleotides, that is bound to 21 proteins. The large subunit is composed of a 5S RNA subunit, 120 nucleotides, a 23S RNA subunit, 2,900 nucleotides, and 31 proteins. 17. Ribosome of E. coli, a bacterium, 18, 962. Ribosome subunit runners are proteins. 70S, 50S, 23S, 2904. NT, 31. 5S, 120 NT. 30S. 16S, 1542. NT. 21. Affinity label for the tRNA binding sites on the E. coli ribosome allowed the identification of A and P site proteins most likely associated with the peptidyl transferase activity. 5. Labeled proteins are L27, L14, L15, L16, L2. At least L27 is located at the donor site, as shown by Ecolets and AP. Chernilovsky. 19, 20. Additional research has demonstrated that the S1 and S21 proteins, in association with the 3' of 16S ribosomal RNA, are involved in the initiation of translation. 21. Archaeal ribosomes, edit. Archaeal ribosomes share the same general dimensions of bacteria ones, being a 70S ribosome, made up from a 50S large subunit, a 30S small subunit, and containing 3 RNA chains. However, on the sequence level, they are much closer to eukaryotic ones than to bacterial ones. Every extra ribosomal protein archaea have compared to bacteria has a eukaryotic counterpart, while no such relation applies between archaea and bacteria. 22, 23, 24. Eukaryotic ribosomes, edit. Main article, Eukaryotic ribosome. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Eukaryotes have 80s ribosomes located in their cytosol, each consisting of a small, 40s, and large, 60s, subunit. Their 40s subunit has an 18s RNA, 1900 nucleotides, and 33 proteins. 25, 26. The large subunit is composed of a 5s RNA, 120 nucleotides, 28s RNA, 4700 nucleotides, a 5.8s RNA, 160 nucleotides, subunits and 49 proteins. 17, 25, 27. Eukaryotic cytosolic ribosomes, are Norvegicus, 18, 65. Ribosome subunit runners are proteins. 80s, 60s, 28s, 4718 nt. 49 5.8s, 160 nt. 5s, 120 nt. 40s 18s, 1874 nt, 33. During 1977, Chernilovsky published research that used affinity labeling to identify TNA binding sites on rat liver ribosomes. Several proteins, including L32/33, L36, L21, L23, L28/29 and L13, were implicated as being at or near the peptidyl transferase center. 28. Plastribosomes and mitaribosomes, edit. Main articles, mitochondrial ribosome and chloroplast ribosomes. In eukaryotes, ribosomes are present in mitochondria, sometimes called mitaribosomes, and in plastids such as chloroplasts, also called plastribosomes. They also consist of large and small subunits bound together with proteins into 170s particle. 17. These ribosomes are similar to those of bacteria, and these organelles are thought to have originated as symbiotic bacteria. 17. Of the two, chloroplastic ribosomes are closer to bacterial ones than mitochondrial ones are. Many pieces of ribosomal RNA in the mitochondria are shortened, and in the case of 5s RNA, replaced by other structures in animals and fungi. 29. In particular, Leishmania tarentuli has a minimalized set of mitochondrial RNA. 30. In contrast, plant mitaribosomes have both extended RNA and additional proteins as compared to bacteria, in particular, many pentatricopeptide repeat proteins. 31. The cryptomonad and chlorarachnophite algae may contain a nucleomorph that resembles a vestigial eukaryotic nucleus. 32. Eukaryotic ATS ribosomes may be present in the compartment containing the nucleomorph. 33. Making use of the differences, edit. The differences between the bacterial and eukaryotic ribosomes are exploited by pharmaceutical chemists to create antibiotics that can destroy a bacterial infection without harming the cells of the infected person. Due to the differences in their structures, the bacterial 70s ribosomes are vulnerable to these antibiotics while the eukaryotic ATS ribosomes are not. 34. Even though mitochondria possess ribosomes similar to the bacterial ones, mitochondria are not affected by. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Figure 4. Atomic structure of the 50S subunit from Halocular Marasmortulae. Proteins are shown in blue and the two RNA chains in brown and yellow. 39. The small patch of green in the center of the subunit is the active site. These antibiotics because they are surrounded by a double membrane that does not easily admit these antibiotics into the organelle. 35. A noteworthy counterexample is the antineoplastic antibiotic chloramphenicol, which inhibits bacterial 50S and eukaryotic mitochondrial 50S ribosomes. 36. Ribosomes in chloroplasts, however, are different. Antibiotic resistance in chloroplast ribosomal proteins is a trait that has to be introduced as a marker, with genetic engineering. 37. Common properties, edit. The various ribosomes share a core structure, which is quite similar despite the large differences in size. Much of the RNA is highly organized into various tertiary structural motifs, for example, pseudoknots that exhibit coaxial stacking. The extra RNA in the larger ribosomes is in several long, continuous insertions, 38, such that they form loops out of the core structure without disrupting or changing it. 17. All of the catalytic activity of the ribosome is carried out by the RNA. The proteins reside on the surface and seem to stabilize the structure. 17. High-resolution structure, edit. The general molecular structure of the ribosome has been known since the early 1970s. In the early 2000s, the structure has been achieved at high resolutions, of the order of a few angstroms. The first papers giving the structure of the ribosome at atomic resolution were published almost simultaneously in late 2000. The 50S, large prokaryotic, subunit was determined from the archaeon Halocular Marasmortulae, 39, and the bacterium Deinococcus radiojurans, and the structure of the 30S subunit was determined from the bacterium Thymus thermophilus. 16, 40, these structural studies were awarded the Nobel Prize in Chemistry in 2009. In May 2001 these coordinates were used to reconstruct the entire T. Thermophilus 70S particle at 5.5A resolution. 41. Two papers were published in November 2005 with structures of the Escherichia coli 70S ribosome. The structures of a vacant ribosome were determined at 3.5A resolution using X-ray crystallography. 42. Then, 2 weeks later, a structure based on cryo-electron microscopy was published, 43, which depicts the ribosome at 1115A resolution in the act of passing a newly synthesized protein strand into the protein-conducting channel. The first atomic structures of the ribosome complexed with tRNA and mRNA molecules were solved by using X-ray crystallography by two groups independently, at 2.8A, 44, and at 3.7A, 45. These structures allow one to see the details of interactions of the Thymus thermophilus ribosome with. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF mRNA and with donors bound at classical ribosomal sites. Interactions of the ribosome with long RNAs containing Shine-Dalgarno sequences were visualized soon after that at 4.55.5a. Resolution 46. In 2011, the first complete atomic structure of the eukaryotic ATS ribosome from the yeast Saccharomyces cerevisiae was obtained by crystallography. 25. The model reveals the architecture of eukaryote-specific elements and their interaction with the universally conserved core. At the same time, the complete model of a eukaryotic 40S ribosomal structure in Tetrahymena Thermophila was published and described the structure of the 40S subunit, as well as much about the 40S subunit's interaction with EIF1 during translation initiation. 26. Similarly, the eukaryotic 60S subunit structure was also determined from Tetrahymena Thermophila in complex with EIF6. 27. Function, edit. Ribosomes are minute particles consisting of RNA and associated proteins that function to synthesize proteins. Proteins are needed for many cellular functions, such as repairing damage or directing chemical processes. Ribosomes can be found floating within the cytoplasm or attached to the endoplasmic reticulum. Their main function is to convert genetic code into an amino acid sequence and to build protein polymers from amino acid monomers. Ribosomes act as catalysts in two extremely important biological processes called peptidal transfer and peptidal hydrolysis. 5.47. The PT center is responsible for producing protein bonds during protein elongation. 47. In summary, ribosomes have two main functions: decoding the message, and the formation of peptide bonds. These two functions reside in the ribosomal subunits. Each subunit is made of one or more RNAs and many are proteins. The small subunit, 30S in bacteria and archaea, 40S in eukaryotes, has the decoding function, whereas the large subunit, 50S in bacteria and archaea, 60S in eukaryotes, catalyzes the formation of peptide bonds, referred to as the peptidal transferase activity. The bacterial and archaeal small subunit contains the 16S rRNA and 21R proteins, Escherichia coli, whereas the eukaryotic small subunit contains the 18S rRNA and 32R proteins. Saccharomyces cerevisiae, although the numbers vary between species. The bacterial large subunit contains the 5S and 23S RNAs and 34R proteins, E. coli, with the eukaryotic large subunit containing the 5S, 5.8S, and 25S/28S RNAs and 46R proteins, S. cerevisiae. Again, the exact numbers vary between species. 48. Translation, edit. Main article, Translation, Biology. Ribosomes are the workplaces of protein biosynthesis, the process of translating mRNA into protein. The mRNA comprises a series of codons which are decoded by the ribosome to make the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF protein. Using the mRNA as a template, the ribosome traverses each codon, 3 nucleotides, of the mRNA, pairing it with the appropriate amino acid provided by an aminosultana. Aminosultana contains a complementary anticodon on one end and the appropriate amino acid on the other. 4. Fast and accurate recognition of the appropriate tRNA, The ribosome utilizes large conformational changes, conformational proofreading. 49. The small ribosomal subunit, typically bound to an aminosultana containing the first amino acid methionine, binds to an August codon on the mRNA and recruits the large ribosomal subunit. The ribosome contains 3 RNA binding sites, designated A, P, and E. The A site binds an aminosultana or termination release factors, 50, 51. The P site binds a peptidyltana, a tRNA bound to the polypeptide chain, and the E site, exit, binds a free tRNA. Protein synthesis begins at a start codon August near the 5' end of the mRNA. mRNA binds to the P site of the ribosome first. The ribosome recognizes the start codon by using the Shine-Dalgarno sequence of the mRNA in prokaryotes and Kozak box in eukaryotes. Although catalysis of the peptide bond involves the C2-hydroxyl of RNA's P site adenosine in a proton shuttle mechanism, other steps in protein synthesis, such as translocation, are caused by changes in protein conformations. Since their catalytic core is made of RNA, ribosomes are classified as ribozymes, 52, and it is thought that they might be remnants of the RNA world. 53. Figure 5, translation of mRNA, 1, by a ribosome, 2, shown as small and large subunits, into a polypeptide chain, 3. The ribosome begins at the start codon of RNA, August, and ends at the stop codon, UAG. In Figure 5, both ribosomal subunits, small and large, assemble at the start codon, towards the 5' end of the mRNA. The ribosome uses tRNA that matches the current codon, triplet, on the mRNA to append an amino acid to the polypeptide chain. This is done for each triplet on the mRNA, while the ribosome moves towards the 3' end of the mRNA. Usually in bacterial cells, several ribosomes are working parallel on a single mRNA, forming what is called a polyribosome or polysome. Co-translational folding, edit. The ribosome is known to actively participate in the protein folding. 54, 55. The structures obtained in this way are usually identical to the ones obtained during protein chemical refolding, however, the pathways leading to the final product may be different. 56, 57. In some cases, the ribosome is crucial in obtaining the functional protein form. For example, one of the possible mechanisms of folding of the deeply knotted proteins relies on the ribosome pushing the chain through the attached loop. 58. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Figure 6, a ribosome translating a protein that is secreted into the endoplasmic reticulum. Addition of translation-independent amino acids, edit. Presence of a ribosome quality control protein RQC2 is associated with MNA-independent protein elongation. 59, 60. This elongation is a result of ribosomal addition, via tanners brought by RQC2, of cattails. Ribosomes extend the C-terminus of a stalled protein with random translation independent sequences of alanines and T-rheanines. 61, 62. Ribosome locations, edit. Ribosomes are classified as being either free or membrane-bound. Free and membrane-bound ribosomes differ only in their spatial distribution, they are identical in structure. Whether the ribosome exists in a free or membrane-bound state depends on the presence of an targeting signal sequence on the protein being synthesized, so an individual ribosome might be membrane-bound when it is making one protein, but free in the cytosol when it makes another protein. Ribosomes are sometimes referred to as organelles, but the use of the term organelle is often restricted to describing subcellular components that include a phospholipid membrane, which ribosomes, being entirely particulate, do not. For this reason, ribosomes may sometimes be described as non-membranous organelles. Free ribosomes, edit. Free ribosomes can move about anywhere in the cytosol, but are excluded from the cell nucleus and other organelles. Proteins that are formed from free ribosomes are released into the cytosol and used within the cell. Since the cytosol contains high concentrations of glutathione and is therefore a reducing environment, proteins containing disulfide bonds, which are formed from oxidized cysteine residues, cannot be produced within it. Membrane-bound ribosomes, edit. When a ribosome begins to synthesize proteins that are needed in some organelles, the ribosome making this protein can become membrane-bound. In eukaryotic cells this happens in a region of the endoplasmic reticulum, called the ruffer. The newly produced polypeptide chains are inserted directly into the by the ribosome undertaking vectorial synthesis and are then explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Transported to their destinations, through the secretory pathway. Bound ribosomes usually produce proteins that are used within the plasma membrane or are expelled from the cell via exocytosis. 63. Biogenesis, edit. Main article, Ribosome biogenesis. In bacterial cells, ribosomes are synthesized in the cytoplasm through the transcription of multiple ribosome gene operons. In eukaryotes, the process takes place both in the cell cytoplasm and in the nucleolus, which is a region within the cell nucleus. The assembly process involves the coordinated function of over 200 proteins in the synthesis and processing of the forerunners, as well as assembly of those runners with the ribosomal proteins. 64. Origin, edit. The ribosome may have first originated as a protoribosome, 65, possibly containing a peptidyl transferase center, PTC, in an RNA world, appearing as a self-replicating complex that only later evolved the ability to synthesize proteins when amino acids began to appear. 66. Studies suggest that ancient ribosomes constructed solely of RNA could have developed the ability to synthesize peptide bonds. 67, 68, 69, 70, 71. In addition, evidence strongly points to ancient ribosomes as self-replicating complexes, where the RNA in the ribosomes had informational, structural, and catalytic purposes because it could have coded fourteners and proteins needed for ribosomal self-replication. 72. Hypothetical cellular organisms with self-replicating RNA but without DNA are called ribocytes, or ribocells. 73, 74. As amino acids gradually appeared in the RNA world under probiotic conditions, 75, 76, their interactions with catalytic RNA would increase both the range and efficiency of function of catalytic RNA molecules. 66. Thus, the driving force for the evolution of the ribosome from an ancient self-replicating machine into its current form as a translational machine may have been the selective pressure to incorporate proteins into the ribosome's self-replicating mechanisms, so as to increase its capacity for self-replication. 72, 77, 78. Heterogeneous ribosomes, edit. Ribosomes are compositionally heterogeneous between species and even within the same cell, as evidenced by the existence of cytoplasmic and mitochondria ribosomes within the same eukaryotic cells. Certain researchers have suggested that heterogeneity in the composition of ribosomal proteins in mammals is important for gene regulation, i.e., the specialized ribosome hypothesis. 79, 80. However, this hypothesis is controversial and the topic of ongoing research. 81, 82. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Heterogeneity in ribosome composition was first proposed to be involved in translational control of protein synthesis by Vince Morrow and Gerald Edelman. 83. They proposed the ribosome filter hypothesis to explain the regulatory functions of ribosomes. Evidence has suggested that specialized ribosomes specific to different cell populations may affect how genes are translated. 84. Some ribosomal proteins exchange from the assembled complex with cytosolic copies. 85. Suggesting that the structure of the in vivo ribosome can be modified without synthesizing an entire new ribosome. Certain ribosomal proteins are absolutely critical for cellular life while others are not. In budding yeast, 14/78 ribosomal proteins are non-essential for growth, while in humans this depends on the cell of study. 86. Other forms of heterogeneity include post-translational modifications to ribosomal proteins such as acetylation, methylation, and phosphorylation. 87. Arabidopsis. 88, 89, 90, 91. Viral internal ribosome entry sites, IRESs, may mediate translations by compositionally distinct ribosomes. For example, 40S ribosomal units without S25 in yeast and mammalian cells are unable to recruit the CRPVIGRIs. 92. Heterogeneity of ribosomal RNA modifications plays a significant role in structural maintenance and forward/or function, and most mRNA modifications are found in highly conserved regions. 93, 94. The most common RNA modifications are pseudoridylation and 2O-methylation of ribose. 95. See also, edit. Aminoglycosides. Biological machines. Post-translational modification. Protein dynamics. Ribosome-associated vesicle. RNA tertiary structure. Translation, genetics. Wobble base pair. Ada Yonath, Israeli crystallographer known for her pioneering work on the structure of the ribosome, for which she won the Nobel Prize. References, edit. 1 Connicott S., February 2016. Dynamic remodeling events drive the removal of the ITS2 spacer sequence during assembly of 60S ribosomal subunits in S. Cerevigi, PhD thesis. Carnegie Mellon University. Archived from the original on 3 August 2017. 2 Weiler U, Nova L., 2008. Allgemeine und Molekuläre Botanik, in German. Stuttgart, Georg. Theumverlag. p. 532. ISBN 9783131527912. 3 De la Cruz J., Karbstein K., Wilford J. L., 2015. Functions of ribosomal proteins in assembly of eukaryotic ribosomes in vivo. Annual Review of Biochemistry. 84, 93-129. Do. 10.1146/ANV. Biochem 060614-033917. PMC 4772166. PMID 257068898. 4 Suitable by nature translation /RNA translation. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 5 ABC Tirumalai Mr. Rivas M. Tran Q. Fox GE, November 2021. The Peptidal Transferase Center, a window to the past. Microbiome Molbiol Revelation 85. 4E0010421 10.1128/MMBR.00104-21. PMC 8579967. PMID 347560086. 6 Ben R. Sloof P. 1987. Evolution of the mitochondrial protein synthetic machinery. Biosystems. 21, 15168. Bibcode, 1987 by ZIS 21, 51b. Do. 10.1016/0303-2647, 87, 90006-2. PMID 2446672. 7 Ribosomes. Archived from the original on 20 March, 2009. Retrieved 28 April, 2011. 8 Pallid GE, January 1955. A small particulate component of the cytoplasm. The Journal of Biophysical and Biochemical Cytology. 1, 15968 do. 10.1083/JCB.1.1.59. PMC 2223592. PMID 143814828. 9 Reinberger, Hans-Georg, September 2022. A Brief History of Protein Biosynthesis and Ribosome. Research. Lindau Nobel Laureate Meetings. Retrieved 16 August 2023. 10 Roberts R.B., ed. 1958. Introduction. Microsomal Particles and Protein Synthesis. New York. Pergamon Press, Incorporated. 11 The Nobel Prize in Physiology or Medicine 1974. Nobelprize.org. The Nobel Foundation. Archived from the original on 26 January 2013. Retrieved 10 December 2012. 12 2009 Nobel Prize in Chemistry. The Nobel Foundation. Archived from the original on 28 April 2012. Retrieved 10 December 2012. 13 Kurland C.G., 1960. Molecular characterization of ribonucleic acid from Escherichia coli ribosomes. Journal of Molecular Biology. 2, 28391 do. 10.1016/S0022-2836, 60, 80029-0. 14 Wilson D.N., Doudna Kate J.H., May 2012. The Structure and Function of the Eukaryotic Ribosome. Cold Spring Harbor Perspectives in Biology. 450115362. 10.1101/perspect.0111536. PMC 3331703. PMID 2255023. 15 Nissen P., Hansen J., Ban N., Moore P.B., Stitz Ta., August 2000. The Structural Basis of Ribosome. Activity in Peptide Bond Synthesis, PDF. Science. 289, 5481 9/2030. Bibcode, 2000 SCI, 289920N. Do, 10.1126/science.289.5481.920. PMID 109379990. S2 SID 83701119. Archived from the original, PDF, on 30 November 2020. 16 AB Wimberley BT, Broderson D., Clements W.M., Morgan Warren R.J., Carter A.P., Von Hin C., Harch T. Ramakrishnan V., September 2000. Structure of the 30S ribosomal subunit. Nature. 407, 6802 327. 39 Bibcode, 2000 Nature.407327W. Do, 10.1038/350300006. PMID 11014182. S2 SID 44199944. 17 ABCDEFG Alberts B., Johnson O., Lewis J., Raff M., Roberts K., Walter P., 2002. Membrane bound. Ribosomes define the rougher. Molecular Biology of the Cell, 4th ed. New York, Garland. Science. p. 342. ISBN 978-0-8153-4072-0. 18 AB Garrett R., Grisham cm. 2009. Biochemistry, 4th ed. Cengage Learning Services. ISBN 978-0-495-11464-2. 19 Kalatsi, Kuchler E., Stoffler G., Chernilovsky A.P., April 1976. The site of reaction on ribosomal protein L27 with an affinity label derivative of tRNA MET-F. Feb's letters. 63, 22836. Do. 10.1016/0014-5793, 76, 80112-3. PMID 770196. 20 Chernilovsky A.P., Kalatsi E., Stoffler G., Kiechler E., January 1974. Proteins at the tRNA binding sites of Escherichia coli ribosomes. Proceedings of the National Academy of Sciences of the United States of America. 71, 1234. Bibcode, 1974 NAS, 71230C. Do. 10.1073/NAS.71.1.230. PMC 387971. PMID 4589893. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 21 Chernilovsky AP, Kurland CG, Stoffology, October 1975. 30 S ribosomal proteins associated with the 3 terminus of 16 sRNA. Feb's letters. 58, 12814 do, 10.1016/0014-5793, 75, 80279-1. PMID 1225593. 22 Cullen KE, 2009. Archaeal ribosomes. Encyclopedia of Life Science. New York, Facts on File. pp. 15. Do. 10.1024/9780470159020000293.pub3. ISBN 9780470015902. S2 SID 2437305776. 23 Tirumalai Mr. Anane Bedico D. Roger Shah, Fox GE, November 2021. Net charges of the ribosomal proteins of the S10 and SPC clusters of halophiles are inversely related to the degree of halotolerance. Microbial. Spectre. 9, 3E01782221 do, 10.1128/spectrum.01782-21. PMC 8672879. PMID 34908470. 24 Wang J. Das Gupta I, Fox GE, the 28th of April 2009. Many non-universal archaeal ribosomal proteins are found in conserved gene clusters. Archaea. 2, 424151 do, 10.1055/2009/971494. PMC 2686390. PMID 19478915. 25 ABC Ben Shemo, Garioda Lubres N, Melnikov S, Jenna L, Yusupova G, Yusupov M. December 2011. The structure of the eukaryotic ribosome at 3.0 A resolution. Science. 334. 6062 1524 9. Bibcode 2011 SCI 334.1524b. Do 10.1126/science.1212642. PMID 22096102. S2 SID 9099683. 26 AB Rebel J, Lebengut M, Atade SF, Hager, Ban N, February 2011. Crystal structure of the eukaryotic 40S ribosomal subunit in complex with initiation factor 1, PDF. Science. 331, 6018. 736. Bibcode 2011 SCI 331730R. Do 10.1026/science.1198308. HDL 20.500.11850/153130. PMID 21205638. S2 SID 247715575. 27 AB Klinges, Voigts Hoffman F, Lebengut M, Arpagaus S, Ban N, November 2011. Crystal. Structure of the eukaryotic 60S ribosomal subunit in complex with initiation factor 6. Science. 334. 6058-9418. Bibcode 2011 SCI 334941K. Do 10.1126/science.12112004. PMID 22052974. S2 SID 206536444. 28 Fabijanski S. Pellegrini M. 1977. Identification of proteins at the peptidyltin binding site of rat liver ribosomes. Molecular and General Genetics. 184, 35516-2, 10.1007/BF00431588. PMID 695020. S2 SID 9751945. 29 Agrawal RK, Shoma Mr. December 2012. Structural aspects of mitochondrial translational apparatus. Current opinion in structural biology. 22, 6797803 10.1016/J.B.2012.08.003. PMC 35136551. PMID 2295947. 30 Shoma Mr. Booth T.M., Simpson L., Marslow Th. Agrawal R.K., June 2009. Structure of a mitochondrial ribosome with minimal RNA. Proceedings of the National Academy of Sciences of the United States of America. 106, 24 9637 42. Bibcode 2009 as 106.9637S. Do 10.1073/NAS.09016311106. PMC 2700991. PMID 194978663. 31 Walls F, Nguyen TT, Arrive M, Beauclerc, Chisher J, Haman P, Kuhn L, Quadrado M, Myro H. Hashem Y, Geige P, January 2019. Small is big in Arabidopsis mitochondrial ribosome. Nature. Plants. 5, 11061172, 10.1038/S41477-018-0339Y. PMID 3062696. S2 SID 58004990. 32 Archibald J.M., Lane C.E., 2009. Going, not quite gone, nucleomorphs as a case study in nuclear genome reduction. The Journal of Heredity. 100, 558292, 10.1093/Jared/ESP055. PMID 196175523. 33 Specialized internal structures of prokaryotes boundless microbiology. Courses.lumenlearning.com. Retrieved 24 September 2021. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 34 Reeked Me, Douthwit S., Pugliese IJD, June 1999. Basis for prokaryotic specificity of action of aminoglycoside antibiotics. The EMBO Journal. 18, 11 3133 8 do 10.1000/embodge/18.11.3133 PMC 1171394 PMID 1035784 35 O'Brien T.W., May 1971. The general occurrence of 55S ribosomes in mammalian liver. Mitochondria. The Journal of Biological Chemistry. 246, 103409 17 do, 10.1016/S0021. 9258, 18, 62239-2. PMID 493061. 36 chloramphenicol-induced bone marrow suppression. JAMA. 213, 7 1183 1184. The 17th of August 1970. Do 10.1011/JAMA.1970.03170330063011. ISSN 0098-7484. PMID 5468266. 37 Newman SM, Boynton J.E., Gillum N.W., Randolph Anderson B.L., Johnson M., Harris A., December 1990. Transformation of chloroplast ribosomal RNA genes in chlamydomonas, molecular and genetic. Characterization of integration events. Genetics. 126, 487588 do, 10.1000/genetics/126.4.875. PMC 1204285. PMID 1981764. 38 Peniv Pi, Fakret Aharaval S, Patel VJ, Cannon JJ, Gutel R, Petrofaz, Williams LD, Glass JB. August 2020. Supersized ribosomal RNA expansion segments in a scarred archaea. Genome. Biology and Evolution. 12, 10 1694 1710 do, 10.1093/GBE/Ivara 170. PMC 7594248. PMID 327856881. 39 AB Ban N, Nissen P, Hansen J, Moore PB, Stitz Ta, August 2000. The complete atomic structure of the large ribosomal subunit at 2.4 resolution. Science. 289, 5481 90520. Bibcode 2000 SCI 289905B. Site CX 10.1.1.58.2271. Do 10.1126/science.289.5481.905. PMID 1093789. 40 Schluens NF, Tassilja, Zaravaka, Harms J, Gluman M, Yane LD, Bashana, Bartels H, Agmani. Franceschi F, Jonathan, September 2000. Structure of functionally activated small ribosomal subunit. At 3.3 angstroms resolution. Cell. 102, 561523 do, 10.1016/S0092-8674, 00000084-2. PMID 110074880. S2 SID 1024446. 41 Yusupov mm, Yusupova GZ, Borkama, Lieberman K, Ernest TN, Kate JH, Nola HF, May 2001. Crystal structure of the ribosome at 5.5 A resolution. Science. 292, 5518 88396. Bibcode, 2001 SCI, 2928883Y. Do, 10.1126/science.1060089. PMID 11283358. S2 SID 395059192. 42 Skworth BS, Borovinskaya Ma, Hao CW, Chang W, V Yasunja Joa, Holt on JM, Kate JH, November 2005. Structures of the bacterial ribosome at 3.5 A resolution. Science. 310, 5749 82734. Bibcode, 2005 SCI, 310827S. Do, 10.1126/science.1117230. PMID 16272117. S2 SID 37382005. 43 Mitra K, Skafitzel C, Sheikh T, Tamar F, Jenny S, Brooks C. L, Ban N, Frank J, November 2005. Structure of the E. coli protein conducting channel bound to a translating ribosome. Nature. 438. 7066 31824. Bibcode, 2005 Nature.438318m. Do, 10.1038/Nature 04133. PMC 1351281. PMID 16292303. 44 Selma M, Dunham cm, Murphy FV, Weichselbaumer, Petri S, Kelly AC, Weir Jr., Ramakrishnan V. September 2006. Structure of the 70S ribosome complexed with mRNA and tRNA. Science. 313. 5795 1935 42. Bibcode, 2006 SCI, 313.1935s. Do, 10.1126/science.1131127. PMID 16959973. S2 SID 9737925. 45 Korostloff, Trakanov S, Loberg M, Nola HF, September 2006. Crystal structure of a 70S. Ribosome complex reveals functional interactions and rearrangements. Cell. 126, 6 1065 77. Do, 10.1016/J.cell.2006.08.032. PMID 16962654. S2 SID 134529115. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 46 Yusupova G, Jenna L, Rees B, Moras D, Yusupov M, November 2006. Structural basis for messenger RNA movement on the ribosome. Nature. 444, 7117 3914. Bibcode, 2006 Nature.444391Y. Do, 10.1038/Nature 05281. PMID 17051149. S2 SID 4419198. 47 AB Specialized Internal Structures of Prokaryotes. Courses.lumenlearning.com. Boundless Microbiology. Retrieved the 27th of September, 2018. 48 Lafontaine, D. Tolave, D. 2001. The Function and Synthesis of Ribosomes. Nat Rev Mol Cell Biol. 2, 7514520 10.1038/350800045 HDL, 1842/729. PMID 11433365. S2 SID 2637106. 49 Savvy Y, Clusty T, April, 2013. The Ribosome as an Optimal Decoder, a Lesson in Molecular Recognition. Cell 153, 2471479. Bib code 2013 APS Mary 46006T. Do 10.1016/J.cell.2013.03.032. PMID 23582332. 50 Cacmus G, Samuel S., September, 2017. Escherichia coli. The Journal of Biological Chemistry. 292, 361513415142 do. 10.1074/JBC.M117.7852238. PMC 5592688. PMID 287434745. 51 Konevga L, Soboleva Ing, Mark No 6, Semenkov YP, Wintermeyer W, Rodnina MV, Katunin 6. January 2004. Purine bases at position 37 of tRNA stabilize codon-anticodon interaction in the ribosomal site by stacking and Mg2+ dependent interactions. RNA 10.19101. Do 10.1261/RNA.5142404. PMC 1370521. PMID 146815888. 52 Rodnina MV, Beringer M, Wintermeyer W, January 2007. How ribosomes make peptide bonds. Trends in Biochemical Sciences. 32, 1/2026 do. 10.1016/J.types.2006.11.007. PMID 171575507. 53 Sec. Tr. August 2000. Structural Biology. The ribosome is a ribozyme. Science. 289. 5481. 878879. Do 10.1016/Science.289.5481.878. PMID 10960319. S2 SID 24172338. 54 Banerjee D, Samuel S., October 2014. Protein folding activity of the ribosome, PFA, a target for antiprion compounds. Viruses. 6, 10 3907 3924 do. 10.3390/V6103907. PMC 4213570. PMID 2534165 55 carat Fedorofen, Baldwin 2, December 1997. Co-translational protein folding. The Journal of Biological Chemistry. 272, 523271532718 10.1074/jbc.272.52.32715. PMID 9407040. 56 carat Baldwin RL, June 1975. Intermediates in protein folding reactions and the mechanism of protein folding. Annual Review of Biochemistry. 44, 14534752, 10.100144.0701750023221. PMID 10949116. 57 carat Das D, Dasso, Samantha D, Ghosh J, Das Gupta S, Bhattacharya, Bajua, Sainul S, Das Gupta C. August 2008. Role of the ribosome in protein folding, PDF. Biotechnology Journal. 3, 8999. 1009 do. 10.1002/biot.200800098. PMID 18702035. 58 carat Dabrowski Tumansky P. Pyajko M. Neuchatel S. Stasiko, Sulkowska J.I. December 2018. Protein knotting by active threading of nascent polypeptide chain exiting from the ribosome exit channel. The Journal of Physical Chemistry B122, 491161611625. Do. 10.1021/x.jpcb.8b07634. PMID 30198720. S2 SID 52176392. 59 carat Branmano, Stuart Ornstein J, Wong D, Larson O, Williams C. C, Lee G. W, Ju S, King D, Shen P. S. Wabezan J, Dunn J. G, Ruskin S, Inadati, Froster, Weissman J. S, November 2012. A ribosome bound quality control complex triggers degradation of nascent peptides and signals translation. Stress. Cell. 151, 5 1042 1054 do. 10.1016/j.cell.2012.10.044. PMC 35349665. PMID 23178123. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 60 carat Defina LQ, Yao Y, Mwakal J, Nameno, Galopio, Decorti L, Doyeno, Malabart C. Savian U C, Chakio, Fromantrosine M, March 2013. CDC 48 associated complex bound to 60S. Particles is required for the clearance of aberrant translation products. Proceedings of the National Academy of Sciences of the United States of America. 110, 13 5046 5051. Bib code 2013 NAS 110.5046D. Do. 10.1073/NAS.12217241110. PMC 36126664. PMID 234796637. 61 Shan PS, Park J, Chin Y, Lee X, Parser War K, Larson MH, Cox J, Cheng Y, Lambertsum, Weissman. J.S., Branman O., Froster, January 2015. Protein synthesis. RQC2P and 60S ribosomal subunits mediate MNA-independent elongation of nascent chains. Science 347, 6217 7578. Bibcode 2015 SCI 347, 75S. Do 10.1026/science.1259724. PMC 44511101. PMID 255544787. 62 Keeley, J. Gutnikoff, R. 2 January 2015. Ribosome studies turn up new mechanism of protein synthesis. Press release. Howard Hughes Medical Institute. Archived from the original on 12 January 2015. Retrieved 16 January 2015. 63 Alberts B., Johnson O., Lewis J., Raff M., Roberts K., Walter P., 2002. Membrane-bound ribosomes. Define the rougher. Molecular Biology of the Cell, 4th ed. New York, Garland Science. ISBN 978-0-8153-4072-0. 64 Kressler, Dieter, Hurt, ed., Babula, Joshen, 2009. Driving Ribosome Assembly, PDF. Biochimica. IT Biophysica Acta, BBA, Molecular Cell Research. 1803, 6673683. Do 10.1016/j.bamker.2009.10.009. PMID 198799002. 65 Dance, Amber, the 28th of February 2023. How did life begin? One key ingredient is coming into view. A Nobel Prize-winning scientists team has taken a big step forward in its quest to reconstruct an early Earth RNA capable of building proteins. Nature 615, 7950 2225 do 10.1038/D41586-023-00574. 4. PMID 368549222. 66 AB Nola HF, April 2012. Evolution of protein synthesis from an RNA world. Cold Spring Harbor. Perspectives in Biology. 4400368 do 10.1101/kshperspect.0036881. PMC 331269. PMID 206105545. 67 Dabza, 1986. Mutant Studies on the Prokaryotic Ribosome. New York, Springer Verlag. 68 Nola HF, Horvath v. Zimnik L., June, 1992. Unusual resistance of peptidyl transferase to protein extraction procedures. Science 256, 5062 1416. Bibcode 1992 SCI 256.1416N. Do 10.1126/science.1604315. PMID 16043115. 69 Nomura M, Mitsushima S, Ozaki M, Trobe P, Lowry C.V., 1969. Structure and function of ribosomes and their molecular components. Cold Spring Harbor Symposia on Quantitative Biology. 34, 4961. Do. 10.1011/1/1969.034.01.009. PMID 49095119. 70 Krupkin M, Matsof D, Tang H, Metz M, Kaleora R, Belousov MJ, Zimmerman E, Bashano, Jonathan. October 2011. A vestige of a probiotic bonding machine is functioning within the contemporary ribosome. Phil. Trans. ASOC. B366, 1582,972 8 10.1098/rstb.2011.0146. PMC 3158926. PMID 2193050. 71 Bose T, Fridkin G, Davidovik C, Krupkin M, Dinger N, Falkovic R, Pelig Y, Agman I, Bashano. Yonato, February 2022. Origin of life. Protoribosome forms peptide bonds and links RNA and protein. Dominated worlds. Nucleic acids res. 50, 418 15 18 28 do. 10.1093/NA/PKK052. PMC 88868871. PMID 3513769. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 72 AB Root Bernstein M, Root Bernstein R, February 2015. The ribosome as a missing link in the evolution of life. Journal of Theoretical Biology. 367, 13158. Do. 10.1016/j.htb.2014.11.025. PMID 255000179. 73 Yaris M, 2002. Primordial Genetics, Phenotype of the Ribocyte. Annual Review of Genetics. 36. 12551. Do. 10.1011/36.anav.genet.36.031902.105056. PMID 124296889. 74 Forter P, Krupovic M, 2012. The Origin of Virions and Virucells, The Escape Hypothesis. Revisited. Viruses, Essential Agents of Life. pp. 4360. Do. 10.1007/9789400748996_3. ISBN 978-94-007-4898-9. 75 Cetanoanal G, Surfer Held MJ, 2013. The Co-Evolutionary Roots of Biochemistry and Cellular Organization Challenge the RNA World Paradigm. Journal of Molecular Microbiology and Biotechnology. 23, 1215277 do. 10.1159/0003465551. PMID 236152003. S2 SID 417252226. 76 carat Saladino R, Botta G, Pino S, Costanzo G, D.I. Moro E, August 2012. Genetics first or metabolism? First. The formamide clue. Chemical Society Reviews. 41, 16 5526 65 do. 10.1039/C2CS35066A. HDL 11573/494138. PMID 226840446. 77 carat Fox GE, September 2010. Origin and evolution of the ribosome. Cold Spring Harbor Prospect. Biol. 2, 90034832, do. 10.1001/kshprospect.0034883. PMC 2926754. PMID 205347711. 78 carat Fox GE, 2016. Origins and early evolution of the ribosome. In Hernandez G, Jagas R, eds. Evolution of the protein synthesis machinery and its regulation. Switzerland, Springer, Cham. pp. 3160. do. 10.1007/978-3-319-39468-8. ISBN 978-3-319-39468-8. S2 SID 274930054. 79 carat Shized, Fuji IK, Kovari Kilometer, Janath NR, Rost HL, Tuyul MN, Bana M, July 2017. Heterogeneous. Ribosomes preferentially translate distinct subpools of MNA's genome-wide. Molecular cell. 67. 1. Elsevier BV, 7183.E7. do. 10.1016/J.Molchel.2017.05.021. PMC 5548184. PMID 286255553. 80 carat XUS, Bana M, May 2012. Specialized ribosomes, a new frontier in gene regulation and organismal biology. Nature Reviews. Molecular Cell Biology. 13, 6. Springer Science and Business. Media LLC, 355369. do. 10.1038/NRM 3359. PMC 4039366. PMID 2261740. 81 carat Ferretti MB, Carbstein K, May 2019. Does functional specialization of ribosomes really exist? RNA. 25, 5. Cold Spring Harbor Laboratory, 521538. do. 10.1261/RNA.069823.118. PMC 6467006. PMID 307333326. 82 carat Farlabons KI, Ogawa LM, Basagar SJ, October 2019. Ribosomopathies, Old Concepts, New Controversies. Trends in Genetics. 35, 10. Elsevier BV, 754767. do. 10.1016/J.Tig.2019.07.004. PMC 6852887. PMID 31376929. 83 Moro VP, Edelman GM, September 2002. The Ribosome Filter Hypothesis. Proceedings of the National Academy of Sciences of the United States of America. 99, 19120316. Bib code 2002 NAS 9912031M. Do 10.1073/NAS.1924424999. PMC 129393. PMID 12221294. 84 XUS, Barner M., May 2012. Specialized Ribosomes, A New Frontier in Gene Regulation and Organismal Biology. Nature Reviews. Molecular Cell Biology. 13, 635569. Do 10.1038/NRM 3359. PMC 40393666. PMID 2261740. 85 Mathis AD, Naylor BC, Carson RH, Evans E, Harwell J, Necht J, Heck ME, Peeler FF, Miller BF. Hamilton KL, Tronstrom MK, Bickman BT, Price JC, February 2017. Mechanisms of in vivo ribosome. Maintenance change in response to nutrient signals. Molecular and cellular proteomics. 16, 2. 243254. Do 10.1074/MCP.M116.0632555. PMC 5294211. PMID 279325527. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 86 Stephen KK, McCormick Ma, Fam Kilometer, Mackay VL, Delaney Jr., Murray Kami CJ, et al. May 2012. Ribosome deficiency protects against stress in Saccharomyces cerevisiae. Genetics. 191, 1. Genetics Society of America, 107118. Do 10.1534/genetics.111.136549. PMC 33385253. PMID 2237760. 87 Lee SW, Berger SJ, Martin Avik S, Pasatolik L, Anderson GA, Shen Y, et al. April 2002. Direct mass spectrometric analysis of intact proteins of the yeast large ribosomal subunit using capillary LC/FTICA. Proceedings of the National Academy of Sciences of the United States of America. 99. 5942 5947. Bib code 2002 NAS 99.5942L. Do 10.1073/NAS.082119899. PMC 122881. PMID 11983894. 88 Carol H, Heselwood JL, Ito J, Miller R, February 2008. Analysis of the Arabidopsis cytosolic ribosome proteome provides detailed insights into its components and their post-translational modification. Molecular and cellular proteomics. 7. 2347369 do 10.1074/mcp.m7000052 MCP 200 PMID 1793414 89 Odint Savati, Muller EC, Ivanov AV, Igorov Ta, Bienata, Vladimirov SN, IT AL. April 2003. Characterization and analysis of post-translational modifications of the human large cytoplasmic ribosomal subunit proteins by mass spectrometry and Edman sequencing. Journal of Protein Chemistry 22, 3249258 10.1023/A10250684196998 PMID 129623225 S2 SID 1071025 90 UY, GH, Doudna J.A., Leary J.A., June 2005. Mass spectrometric analysis of the human 40S. Ribosomal subunit, native and HCV IAS-bound complexes. Protein Science 14, 6 1438 1446 do 10.1110/PS.0412930005 PMC 2253395 PMID 158831884 91 Zidane Q, Wang Z, De Mayo, Hart GW, June 2010. Oboknak cycling enzymes associate with the translational machinery and modify core ribosomal proteins. Molecular biology of the cell. 21, 12. 1922-1936. Do 10.1091/mbc.e09-11-0941. PMC 2883937. PMID 2041038. 92 Landry DM, Hertz Me, Thomson SR, December 2009. RPS 25 is essential for translation initiation by the Dysostroviridae and hepatitis C viral IRESs. Genes and development. 23, 2753 2764. Do 10.1101/GAD.18322209. PMC 2788332. PMID 199521110. 93 Decatur WA, Fournier MJ, July 2002. RNA modifications and ribosome function. Trends in Biochemical Sciences. 27, 734451 do 10.1016/S0968-0004, 02, 021009-6. PMID 121140023. 94 Nagia SK, Musnikov AG, Kratsat H, Hayes Marnai, Klar-Holes BP, November 2017. Visualization of chemical modifications in the human ATS ribosome structure. Nature. 551, 7681 472477. Bibcode 2017 Nature.551472N. Do 10.1038/Nature24482. PMID 2914318. S2 SID 4465175. 95 GOH, August 2018. Specialized ribosomes and the control of translation. Biochemical Society. Transactions 46, 4855869 10.1042/BST20160426. PMID 299866937. S2 SID 5160977. External links, edit. Lab computer simulates ribosome in motion. Role of the ribosome, Gwen V. Childs, copied here. Ribosome in Proteopedia, the free, collaborative 3D encyclopedia of proteins and other molecules. Ribosomal proteins families in XSpace archived 30 April 2011 at the Wayback Machine. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Molecule of the Month archived 27 October 2009 at the Wayback Machine. Copyright symbol RCSB Protein Data Bank. Ribosome archived 14 November 2010 at the Wayback Machine. Elongation factors archived 16 March 2011 at the Wayback Machine. Pallid. 3D electron microscopy structures of ribosomes at the M-Data Bank, EMDB. This article incorporates public domain material from Science Primer. NCBI. Archived from the original on 8 December 2009. Categories: Ribosome, ribozymes, protein biosynthesis. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.